Exploring Life, Chapter 7. Prokaryotes, Part 1. For the first time in this course, we turn our attention to fully living organisms. Until now, we've explored precellular structures, protobionts, that stood at the threshold of life. One particularly successful protobiont, which developed the ability to replicate, metabolize, and evolve, likely became what scientists refer to as the universal ancestor. This transition, from non-living chemistry to the first true life, may have taken hundreds of millions of years after Earth's formation, likely occurring around 3.5 billion years ago or earlier. All modern life traces back to this early form. From this single ancestor, life diversified. All life forms can be divided into two major categories based on cellular organization: prokaryotes and eukaryotes. This distinction is based on cell structure, and it's one of the most fundamental in biology. Prokaryotes are organisms whose cells lack a nucleus and other membrane-bound organelles. They are always unicellular, and include members of the domains Bacteria and Archaea. Eukaryotes, in contrast, have cells with a true nucleus and membrane-bound organelles. They can be unicellular or multicellular, and all belong to the domain Eukarya. Prokaryotes and eukaryotes represent a fundamental level of classification based on cell structure. But taxonomy begins there and goes much further. Taxonomy is the science of classifying organisms, but it's more than just naming things. It reflects evolutionary relationships, showing how species are connected through common ancestry. At the top of this system are the highest taxonomic ranks, known as domains. These domains represent the earliest evolutionary splits in the history of life. The three domains, Bacteria, Archaea, and Eukarya, form the deepest branches on the tree of life. Both Bacteria and Archaea are prokaryotic, while all eukaryotic life belongs to the domain Eukarya. The diagram on the right shows the three domains of life, all branching from a shared universal ancestor. Archaea are prokaryotic microorganisms often found in extreme environments like hot springs, deep-sea vents, and salt lakes. Bacteria, also prokaryotic, are incredibly diverse and inhabit nearly every environment, from soil and water to our own bodies. Eukarya includes all organisms with eukaryotic cells, those with a nucleus and membrane-bound organelles, such as protists, fungi, plants, and animals. Prokaryotes, Archaea and Bacteria, represent the oldest branches on the tree of life. Notably, Eukarya is more closely related to Archaea than to Bacteria, supporting the idea that eukaryotic cells likely evolved from an ancestral archaeal lineage. During this lecture, we'll focus on prokaryotes, the oldest and most abundant life forms on Earth. They've existed for billions of years, thriving in environments ranging from the everyday to the extreme, from hot springs and salt flats to deep ocean vents. Prokaryotes outnumber and outweigh all other life forms combined, and their success offers vital clues about how life began and adapted on our planet. Understanding these resilient organisms is key to understanding the earliest chapters of life on Earth. Archaea and Bacteria are the two domains that make up the prokaryotic category of life. Let's begin by exploring domain Archaea. Archaea are single-celled microorganisms classified as prokaryotes, meaning they lack a nucleus and other membrane-bound organelles. They represent one of the three primary domains of life, alongside Bacteria and Eukarya. Archaea are remarkable for their ability to survive in some of the most extreme environments on Earth. They thrive in places with extreme temperatures, high acidity, or intense salinity, such as scorching hot springs, acidic pools, and deep-sea hydrothermal vents. Because of these harsh habitats, and because archaea are microscopic, they are rarely encountered directly by humans. Yet despite their invisibility, they are surprisingly widespread, with many species living in common environments like soil, oceans, and even the human gut. Their resilience and adaptability offer important clues about the potential for life in extreme conditions, on Earth and possibly beyond. The thermal features shown in this photo from Yellowstone National Park are famous for their vivid, almost surreal colors, and the photo is not enhanced. These unnaturally bright hues are real, caused by archaea and other microorganisms thriving in the hot springs. Different species inhabit specific temperature zones, forming distinct bands of color. Each zone reflects a unique microbial community, finely tuned to the heat and chemistry of that part of the spring. These stunning gradients aren't just beautiful, they demonstrate how life adapts to extreme conditions, with temperature directly shaping biodiversity in these remarkable environments. Archaea play a vital role in ecosystems, despite often being overlooked. They participate in major biogeochemical cycles, including the carbon and nitrogen cycles, helping to maintain Earth's environmental balance. Their ability to thrive in extreme environments also makes them essential for studying life's adaptability, and they offer valuable insights into the possible origins of life. By understanding archaea, we gain a deeper perspective on both modern ecosystems and ancient biology. Although archaea and bacteria are both prokaryotic, meaning they lack a nucleus and membrane-bound organelles, they differ in several important ways. First, their genetic makeup is distinct. Archaea possess unique genetic sequences that set them apart from bacteria. Second, their cell membranes differ in composition. Archaea have specialized lipids that make their membranes more stable in extreme environments. And third, their evolutionary histories are separate. While they may look similar under a microscope, archaea and bacteria represent two ancient, independent branches of life. There is growing evidence of a potential evolutionary link between archaea and the origin of eukaryotic cells. Some archaeal lineages share key genetic and molecular features with eukaryotes, suggesting a possible connection in early evolution. However, there is still no definitive proof that archaea are the exclusive source of eukaryotic cells. The exact pathway remains an active area of research, with several competing hypotheses. What's clear is that understanding archaea is crucial for piecing together the deep evolutionary history of complex life. The second domain of prokaryotes is bacteria, a vast and diverse group of single-celled organisms. Bacteria make up the largest group of prokaryotes and are the most numerous organisms on Earth, playing vital roles in ecosystems all over the planet. They thrive in a wide range of habitats, especially in soil, where they help break down dead matter and recycle nutrients, essential work they've been doing long before humans appeared. And remarkably, your body may host more bacterial cells than human cells, many of them crucial for digestion, immunity, and overall health. They're also among the oldest life forms, and after billions of years, they're still thriving. Not bad for something you can't even see without a microscope. So, what makes bacteria so special? All bacteria are unicellular, but they come in a variety of shapes that can be seen under a microscope. These shapes help scientists identify and classify different bacterial types. The most common shapes include cocci, spherical in form, center photo. Bacilli, rod-shaped, left photo. Spirilla or spirochetes, with a spiral or corkscrew shape, right photo. It's especially easy to spot the spiral shape when you compare them to rod-shaped bacilli. These structural differences often relate to how bacteria move, group, or interact with their environment. This diagram shows the basic structure of a bacterial cell, highlighting the key components found in all prokaryotic organisms. At the center is the nucleoid, an irregularly shaped region that houses the cell's chromosomal DNA. Unlike eukaryotes, this genetic material is not enclosed by a membrane. Scattered throughout the cytoplasm are ribosomes, the structures responsible for building proteins from genetic instructions. Surrounding the cell is the cell membrane, which controls what enters and exits, and just outside that is the cell wall, which provides structural support and protection. The absence of a nucleus is one of several key features that distinguish bacterial cells from eukaryotic cells. Instead, their genetic material is located in a region called the nucleoid, an area that contains a single, circular DNA chromosome, not enclosed by a membrane. In addition to this main chromosome, many bacteria also carry plasmids, small, circular DNA molecules that are separate from the chromosomal DNA. Plasmids often carry genes that give bacteria special advantages, such as antibiotic resistance or the ability to digest unusual substances. Unlike eukaryotic cells, bacteria do not contain any membrane-bound organelles within their cytoplasm. However, they do have ribosomes, essential molecular machines responsible for protein synthesis. These ribosomes are smaller than those found in eukaryotes, but they carry out the same crucial function. Translating genetic instructions into proteins the cell needs to grow, survive, and reproduce. Most bacteria have a cell membrane surrounded by a cell wall, a rigid structure that gives the cell its shape and mechanical support. The cell wall protects bacteria from harsh environmental conditions and helps prevent the cell from bursting in hypotonic environments. It also serves as a defense mechanism, offering protection against threats like immune system attacks or antibiotics. In some species, the cell wall is further coated with a capsule or slime layer, which provides extra protection, helps bacteria adhere to surfaces, and can even evade immune detection. Some bacteria have a remarkable survival strategy. They form endospores, highly durable structures that allow them to withstand extreme conditions. An endospore is enclosed in a tough, protective coat that resists heat, drying out, radiation, and even chemical damage. In this dormant state, the bacterium can survive for decades, or even centuries, until conditions become favorable again. When the environment improves, the endospore can reactivate, allowing the bacterial cell to resume growth and reproduction. This ability makes endospore-forming bacteria some of the most resilient organisms on Earth. Although bacteria are microscopic, many are highly efficient at moving and interacting with their environment. Some can travel over 100 body lengths per second, far faster, proportionally, than a Formula One car, which covers about 20 body lengths at top speed. They achieve this using flagella, long, whip-like naked protein extensions that rotate like propellers, allowing the cell to swim through liquids. Other bacteria use pili, shorter, hair-like structures that help them stick to surfaces, attach to other cells, or even form bridges to exchange genetic material. Some bacteria also move by secreting slime, allowing them to glide across surfaces or migrate within biofilms, dense communities of microorganisms. Together, these adaptations help bacteria navigate, colonize, and survive in a wide range of environments. Bacteria reproduce through a process called binary fission, a simple and efficient method of asexual reproduction. In this process, the bacterial cell duplicates its DNA, then divides into two identical daughter cells. Each new cell receives an exact copy of the original chromosome, making them genetically identical to the parent. These daughter cells then grow and repeat the cycle, allowing bacterial populations to multiply rapidly under the right conditions. It's a fast, reliable way to expand, and part of what makes bacteria so successful. This diagram illustrates binary fission, the process by which bacteria reproduce. The DNA replication, producing two identical copies of the bacterial chromosome. The cell then elongates, with each copy moving to opposite ends of the cell. Next, the membrane pinches inward, dividing the cell into two identical daughter cells, each ready to start the process again. Bacteria may reproduce asexually, but they adapt quickly through powerful methods of gene exchange. In transformation, they absorb free DNA from their surroundings, a process that may resemble how early protobionts took in molecules from their environment. In conjugation, DNA is transferred directly between cells, often via plasmids. These methods allow for rapid genetic recombination, driving evolution and boosting survival, even without sexual reproduction. Sometimes, bacteria don't go it alone, they team up. Biofilms are dense communities of bacteria that form on surfaces, from river rocks to your teeth. The cells in a biofilm release chemical signals to communicate with one another, attracting more bacteria and coordinating their behavior. Together, they build a kind of protective matrix, allowing them to stick to surfaces and resist threats like antibiotics or the immune system. Biofilms are behind common infections, like ear infections, urinary tract infections, and, of course, dental plaque. In Part 2, we'll continue exploring what makes bacteria so special. Focusing on their incredible metabolic diversity, their major role in shaping Earth's early atmosphere, and the many ways humans rely on them today, in health, food, agriculture, and beyond.